Assalamualaikum and good morning. This is video lab practical class 1. The title is Fish Decisions, Observations and Perservations from group 3. Let me introduce our team from group 3. Number 1 is Elena binti Lim, Maswe Martin, Siti Nordana binti Isnil, Jivia Clayton Balang, Christina Libya Suntai anak Imai. We have 5 people in our team. Hello, my name is Elena binti Lim. Today I'm going to explain about objective. Objective number 1 is able to understand the anatomy of fish and research study the internal and external anatomical structure of fish. Number 2 is make it easy to determine abnormality and understand how disease affect fish anatomy. Number 3 is identify pathogens such as bacteria, virus, fungus, and parasite that cause disease in fish. And the last one is studying biology responses to infections and other health conditions.
For the material that we have used is a dissection kit. Use it to dissect the fish, ethanol 70% and 99%, and to look into our sample we are using a light microscope, and we also use the distilled water for the sample bottle, alcohol lamp to heat up the bladder before dissecting the fish, and dissecting tray. We also used a glass slide and cover slip for the light microscope, and the dropper is to take the distilled water from the bottle to put into the sample bottle, and we also use the tissue. During the lab, we also use a glove to prevent our hand to directly contact to the fish and the alcohol. And the last one is we are using the spotted sardinella fish for the lab.
I'm Elena Lim, so I'm going to talk about fish decisions, where I'm going to talk about procedure. Number 1 is put the fish into dissection tray. If the fish is still alive, euthanize the fish before dissection. Number 2 is observe external of the fish. Number 3 is if there are abnormality, collect the sample for observations under microscope or further investigation. Number 4 is disinfect the fish using the ethanol 70%. Number 5 is dissect the fish to expose its body cavity. Number 6 is observe the fish internal organ. And the last one is isolate those organs. So this is the result when we fish external filter. Number 1, you can see our fish head is normal. Tail or caudal fin is normal. Dorsal fin is normal. And pectoral or pelvic fin is also normal. The eyes of fish is normal. After that, as you can see, the anus is normal, but the skin have a deformation that is abnormal, and the gill is normal. The last one is mouth. The mouth of the fish that we see from external is normal. That is for me. Thank you.
Hello, my name is Siti Nordana binti Isnil, BS 221605551. Today I will be discussing the internal feature of fish, focusing on their vital organs. Fish, being aquatic vertebrates, have fascinating internal structure that enable them to thrive in their environment. And let's start about the liver. The liver in fish plays a crucial role in metabolism, detoxification, and the production of various essential protein, and enzymes. It aids in digestion by producing bile, which helps in the breakdown of fats. In our specimen, the liver appears normal and in good condition, indicative of a healthy fish. Next up is the spleen. The spleen in fish serve multiple functions, including immune response, blood filtration, and storage of red blood cells. A healthy spleen contributes to overall well-being and immune competence of the fish. Our specimen shows a spleen that is normal and function and optimally. Moving on the intestines. The intestines of fish are responsible for nutrient absorption and digestion. They play a crucial role in breaking down food into smaller molecules that can be absorbed by the body. Examination of our specimen reveals intestines that are healthy and functioning efficiently. Now let's talk about the kidney. The kidneys in fish regulate water balance, remove waste product from the blood, and help maintain electrolyte balance. Healthy kidneys are essential for proper osmoregulation and overall metabolic function. Our examination indicates that the kidneys of our fish specimen are normal and in good condition. And the last is the brain. The brain in fish controls various physiological and behavioral function, including sensory perception, movement, and feeding. It is a complex organ that ensures the fish can navigate its environment and respond to stimuli effectively. Our specimen exhibits a healthy and well-developed brain, indicative of its overall health. That's all for me. Thank you.
Hello, my name is Masque Martin and I will be presenting wet mount slide technique. The what and or why. First, the what. In order to accurately look at a specimen under a microscope, a wet mount is made by placing a fluid solution on a slide, suspending a specimen in a solution, and then covering the specimen and the solution with a cover slide. Why? First, it is to increase the specimen translucency and to make it easier to stain. And secondly, using a wet mount slide has the tendency to flatten the specimen, making it easier to view. Common materials needed to perform a wet mount slide include slides, cover slips, cell specimens, fish specifically, scissors, forceps, eyedropper, distilled water, and a compound microscope. Lastly, the procedure. Firstly, you will want to obtain the desired specimen for analysis. Carefully use forceps to hold part of the specimen and using scissors to cut tiny part. Secondly, using forceps to place it on the center of a clean slide. Thirdly, put a drop of distilled water using eyedropper. Fourth, slowly cover it with a cover slip to prevent air bubble from forming. And lastly, place under a microscope and observe. That's all from me. Thank you very much.
Okay, for the tissue preparation, we have 6 procedure. First, we need clean the fish. Second, we need to clean the dissecting set using ethanol 70%. And number 3, we dissect the desired organ carefully. For the organ, we use fish eyes. And number 4, transfer the tissue into buffered formalin 10%. And number 5, after 24 hours, transfer the tissue into boiling solution. And for the last, keep the tissue in the room temperature.
Assalamualaikum and good morning. This is our video lab practical 3 technique microbiology strict pellet method present by group 3. Hello, my name is Elena binti Jimritnim. Today I'm going to talk about material that we use from lab 3. They have 5 material. Number 1 is plate. Number 2 is glass slide. Number 3 is parafilm strip. Number 4 is plantarum P20. And the last one is inoculating needles.
The objective of straight plate method, number one, is to obtain a pure culture of bacteria from a mixed culture. Number two, to obtain well isolate colonies. And number three, to propagate bacteria.
Hello, my name is SQ21403 and I will be explaining the procedure for strict plate method. After all the material are prepared, the inoculating loop was held like a pencil and sterilized by flaming it with a Bunsen burner until it glows red. If using disposable inoculant loop, however, make sure to avoid contact with any surface and only open it when about to be used. Second step will be the loop was cooled for a few seconds. Third step, the loop tip was used to make a colony of bacteria or broad culture. Fourth step, the media agar was taken out and only the lid was lifted enough to perform streaking gently. The tip of the inoculating loop was glided back and forth across the agar surface. Again, very gently. Step 6, the inoculating loop was reflamed and the loop was cooled before use, or if using a disposable, using a new disposable inoculation loop. Step 7, touch the previous streak and repeat the streak in the remaining quarter of the plate. Step 8, repeat step 6 and 7. Step 9, the plate was sealed with a parafilm and the plate was placed upside down in an incubator for about 24 to 72 hours. Additional precaution is to always wear a glove which has been sterilized with ethanol, wear a mask or try not to breathe down on the agar. If possible, only open the agar lid partially to prevent further contamination of other bacteria of substance. That's all from me. Thank you very much.
Hello, I'm Siti Nordana binti Isnil BS221605551. For result strict plate method, we finding reveal that following a 24 hours incubation period at 34 degrees, bacterial proliferation was evident in the initial row atop the agar surface. This observation signifies the adaptability and viability of the bacteria within the sample under the specified temperature and growth medium condition. That's all. Thank you.
For the conclusion is the strict plate method worked well in this experiment for separating bacterial colonies and producing pure cultures, distinct colonies with a range of morphology, size, and colors were observed on the strict plates, demonstrating the effective isolation of many bacteria species. Numerous types of bacteria were successfully isolated from the original sample because to the technique ease of use and adaptability, even with some obstacles like fluctuation in colony growth rate and sporadic contamination. Carefully monitoring and repeated streaking made it possible to isolate pure culture feed for additional analysis. And that's all for me. Thank you very much.
Assalamualaikum and good morning. This is our video lab practical class 4. That is gram staining presented by group 3.
The material of gram staining we use is glove, marker, tissue, bacteria culture, water, Bunsen bubbles, inoculate loop, ethanol 70%, crystal violet iodine solution, suffering, discolyzer, emulsion oil, and light Microsoft. And for this slide is the picture of the material.
Hello, my name is Elena binti Jinuddin. Today I'm going to talk about objective. In lab 4, they have a 4 objectives. Number 1 is able to identify gram staining differently bacteria. Number 2 is accurately identify bacteria pathogen, ensure effective treatment. And number 3 is able to understand basic microbiology technique in veterinary microbiology and aquaculture program. Objective the last one is help to understand the classifications and diagnosis of bacteria.
Hello, I'm Siti Nordana binti Ismail, BS221605551. For gram staining procedure, the first is prepare a clean free glass slide and mark the center area of the slide using a marker pen, and then sterilize an inoculating loop using a Bunsen burner. If using bacteria from an agar plate, add a drop of distilled water to the marked region on the slide using a dropper. And take an inoculum of bacteria and mix it with distilled water on the slide. And if using bacteria from a liquid culture, transfer a drop of the bacterial culture to the marked glass slide and spread the bacteria. Then allow the sample to air dry before staining. And for staining, the first is pour crystal violet and leave it for 60 seconds, then rinse with tap water. Stain with iodine for 60 seconds and wash off with tap water, and tilt the slide at 45 degree angle and add a few drops of alcohol before rinsing again with tap water. Then add saffronin for 60 seconds and wash off with tap water. And the last is allow the sample to dry and observe it under a light microscope. That's all. Thank you.
Hello, my name is Masque Martin and I will be discussing the result of the gram staining. The result of the gram staining yield appearance of the bacteria observed having a somewhat bluish purple color. The result indicate based on the gram staining test to be gram positive, meaning the cell wall having a thick layer of peptidoglycan, about 90% of the cell wall, a set of substance that form the cell walls of many bacteria. If the color appear red or pink under the gram stain, the result will be gram negative, meaning the bacteria have thin layer of peptidoglycan, about 10% of the cell wall, and contain high lipid, fatty acid content. Example of gram-positive bacteria is Lactobacillus, Actinomyces, Bacillus, Clostridium, Corynebacterium, Staphylococcus, and Streptococci. Example of gram-negative bacteria is Acetobacter, Chlamydia, Borrelia, Bordetella, Burkholderia, Enterobacter, Escherichia, Helicobacter, Klebsiella, and Neisseria. That's all from me. Thank you very much.
For the conclusion is when it comes to distinguishing bacteria according to the makeup of their cell walls, the gram staining method was proven to be an invaluable resource. Gram-positive and gram-negative bacteria were distinguished from one another by their unique morphologies and colors through close examination of stained samples. The core ideas of gram staining, such as differences in the uptake of saffronin and crystal violet by the bacterial cells, were effectively illustrated by the experiment. Gram-negative bacteria show pink after counter-staining with safranin because of their thin peptidoglycan layer and outer membranes, while gram-positive bacteria maintains the purple blue of crystal violet because of the thick peptidoglycan layer. Thank you. That's all for me.